This is Unit 5, Part 3. We're continuing with physiology of respiration. Henry's law is another important basic physical-chemical characteristic of gases. When a mixture of gases is in contact with a liquid, water, in your lungs, each gas will dissolve into the liquid in proportion to its partial pressure. And the amount also depends on its solubility. So the key things are the individual partial pressure and its solubility in the liquid, which again, in the case of the lungs, is water. Now, not only is there different partial pressures, but the solubilities in water vary significantly. Carbon dioxide is quite soluble within water, whereas oxygen is not particularly soluble. It's only 1/20th as soluble as carbon dioxide. And nitrogen is practically insoluble in water-based medium. It says in plasma, but we can call it water or plasma. So it's not going to diffuse across and be present within the blood plasma. So the key gases that are moving between the alveolar space and the blood one way or the other are carbon dioxide, CO2, and oxygen, O2. Okay. Again, when we look at atmospheric air, that mixture of gases, it's mostly oxygen and nitrogen. The alveoli contain more CO2 for the reasons I mentioned earlier and more H2O, but less oxygen than air. And these differences, it's funny it's not there. Anyhow, oxygen is lower. So we'll write it in. Sorry. The alveoli contain less oxygen than the air, outside atmospheric air here. Differences being due to diffusion from the alveoli into the blood and diffusion of CO2 from the blood into the alveoli. Humidification of air occurs in the conducting passageways, the nose, the mouth, the pharynx, the trachea, the bronchioles, all the way down as far as the terminal bronchioles. And mixing of these, differing gas from atmospheric and alveolar occurs with each time a person breathes in and out. External respiration involves pulmonary gas exchange. So the factors whatever factors influence movement of O2 and CO2 across the respiratory membrane, you remember I described the respiratory membrane in the lab, and you learned about it last year. It's a very thin membrane made up of the alveolar epithelial, type 1 epithelial cell, and the endothelial cell lining the capillaries, and the fused basal membranes or basal lamina between the two. Key factors influencing movement of O2, in other words, diffusion of O2 and CO2 across the respiratory membrane, are the partial pressure gradients of each one and the solubility in water of each one. The second one is ventilation perfusion coupling, which is a physiological process that optimizes alveolar ventilation and pulmonary blood perfusion, and I'll talk more about that in the next few slides. And then the structural characteristics of the respiratory membrane. These are the key factors that influence movement or diffusion of these gas of O2 and CO2. In terms of the first one, the partial pressure of gas is indicated in this way with P for pressure and then the name of the gas as a subscript. So the partial pressure of oxygen here in venous blood is 40, a number you need to know and remember. And as we mentioned earlier, the alveolar PO2 is 104. Okay. So that means that there's quite a steep pressure gradient, a difference between the alveolar PO2 and the PO2, partial pressure of oxygen, of the venous blood as it comes into the lungs. If this is an alveolus and there's another alveolus and say there's another one in this cluster of alveoli, they are surrounded, of course, by capillaries that snake across the surface. These, say, are in cross section. There's one in longitudinal section. So there's all kinds of capillaries coming across. And where the blood comes into the capillary, the venous PO2 is 40. Whereas in the alveolar space, that means in here, and here, and here, the PO2 is 104. So there's a steep, there's a big difference in the pressure gradient, and therefore that helps drive or pull oxygen, that helps allow oxygen to diffuse into the blood. Now in this graph, and I always recommend that when you look at a graph, you look at the axes first. This is time across the horizontal axis. It's from the time that, average time that blood is in the capillaries from the starting part, the venous side of the capillary, to the end of the capillary, the arterial side of the capillary where it joins the arteries. And this is PO2. You start at 40, right there. The PO2 in the blood is at 40. Okay, on this end. And it goes in and it ends up being, this is not 100, it's not 100%, it's not percentage, it's PO2, and it ends up at 104 in balance with the atmospheric, or the, well, not atmospheric, in balance with the alveolar partial pressure of oxygen. Now notice it only takes 0.25 seconds for the, oh, and by the way, 104 is fully saturated. The blood now is fully saturated with oxygen. All the hemoglobin has as much oxygen as it can take up. And so it only takes 0.25 seconds of the blood being in the capillary, of the capillaries surrounding the alveoli in the lungs, for it to become fully saturated with oxygen. That means if for whatever reason there's some problem and the transfer of oxygen is inhibited, maybe the respiratory membrane got thicker or there's less oxygen in the air or whatever, but you know, so it takes longer for it to diffuse across. Even if it doubles or triples, it will reach, if it doubles, it'll be something, you know, it would go something like this, right? If it tripled in the time it took, then it would go something like this, right? So you have reserve. There is a certain reserve there present, which, you know, can compensate for that. Even if the blood moved, or let's say the blood is moving faster, it's not a question of a disease process or lack of oxygen in that air. The blood is moving faster through the capillaries, and that can happen with increased pressure and force with movement of the blood with exercise. then even if it's moving 3 times faster, it'll still get fully oxygenated by the time it still will, if it moves 3 times faster, it'll still get fully oxygenated because it will be oxygenated so quickly. Okay, partial pressure of in terms of the CO2. Remember, the O2 partial pressure was 40 in the venous blood and 104 in the alveolar gas, the space in the alveoli, and also therefore in the arterial blood. And the CO2, the partial pressure of CO2 in the venous blood is 45, and the alveolar CO2 is 40. The pressure gradient is smaller, but the solubility is much greater, so that compensates for that smaller pressure gradient. Remember, the pressure gradient is an important part of the driving force, but so is the solubility of the gas in water. So much lower partial pressure gradient, but 20 times more soluble than oxygen, so it can easily move across. It should really, I mean, we're talking about water, that's the solute. CO2 moves in equal amounts across as the oxygen diffuses into the blood. CO2 is moving out into the alveolar space. And so you end up with these nice numbers that seem to work quite well. CO2 is 40 and PO2 is 104 in the alveolar space. And here you see a stylized diagram showing the circulatory system. We're talking about blood returning through the venous return to the right side of the heart. This is the right side. There's the right atrium. There's the right ventricle. It gets pumped up to the lungs and goes into the capillaries here surrounding the alveoli, and then returns via the these are pulmonary these are pulmonary arteries, by the way. Arteries carry blood away from the heart, so that's a pulmonary artery. This is just a second. I just wanted to point out that there are 2 circuits. This is the pulmonary circuit, that's a P, and this is the systemic circuit of the circulation. So the blood goes back to the heart through the venous return, and then into the right ventricle, and then it gets pumped up through the pulmonary arteries. You see here, pulmonary arteries up to the lungs, and it goes into the capillaries surrounding the alveolus, and then comes back through the pulmonary veins, returning blood, fully oxygenated blood, back to the heart, to the left atrium, to the left ventricle, and then out into the systemic circulation, and eventually to capillaries in the tissues, where it dumps oxygen into tissues throughout the body and picks up CO2 and returns that to the heart, and the circuit is completed going round. Now, in terms of the gases and partial pressures, let's just clean this up. Okay, now in terms of the partial pressures, as I told you before, PO2 is 104 in the alveolar space, PCO2 is 40. You get gas exchange, oxygen going into the blood and CO2 coming out of the blood. So the blood leaving the alveolar capillaries in the pulmonary veins and going back to the heart will have a PO2 of 104 and a PCO2 of 40, just as it did in the alveolar space. And then it gets pumped around the body, so it's being transported through there's no gas exchange, gas exchange only occurs in capillaries. So it gets pumped around, delivered back to the heart, and delivered out from the heart through the aorta into all the tissues. So it's carrying blood that has a PO2 of 104 and a PCO2 of 40. And then at the tissue level, when it enters into the capillaries and systemic circulation, it releases, the hemoglobin will release oxygen, and the oxygen drops below 40. Well, let's leave it at 40. Or it could be lower than 40 if the tissue is a very active tissue, muscle that's contracting and relaxing at a high rate. PCO2 will be 45 or greater because there's a movement of CO2 from the cells, the tissue, into the blood. And then it travels, it's at these concentrations and travels back to the heart and gets pumped up to the lungs, PO2 40 and PCO2 45. Now, I want to remind you what the body does with that oxygen in the tissues. The cells need to take up oxygen from the blood in the tissue in order to carry out aerobic respiration. Up here, we're talking about external respiration, and here we're talking about internal respiration. And in here, we're talking about the respiration, cellular respiration, which involves generation of ATP, energy in the form of ATP. So this is cellular respiration. It's aerobic respiration using oxygen. Oxygen, of course, is the final electron acceptor in the electron transport chain. That's how cells produce energy through the action of the aerobic respiration, the electron transport chain, and chemiosmosis. The CO2 comes from the breakdown of carbohydrates, particularly glucose, in that process. And so CO2 is being generated. It's oxidation of carbohydrates, oxidation particularly of glucose, and so CO2 builds up as it gets released from that from the oxidation of carbohydrate and goes into the blood, and that gets back to the heart and up to the lungs and pumped out. So 104 for oxygen and 40 for CO2, and then down here it changes to 40 for oxygen or lower and 45 for CO2 or higher. Higher CO2 if you're very, very active, the cells are very active, generating a lot of energy. Okay, let's talk about the second major factor that controls external respiration. That's ventilation perfusion coupling. Both ventilation, which means movement of air into the lungs, and perfusion, which is movement of blood through the lung vasculature, through the capillaries and surrounding alveoli, are tightly regulated relative to each other. The changes in the partial pressure of oxygen inside the alveoli will cause a change in arteriolar diameter. So the arterioles, just before the capillaries surrounding the alveoli, can change in their diameter. And as you know, changes in diameter will cause changes in flow. If it's narrower, if you have vasoconstriction and it gets narrower, then there's less flow, less perfusion of blood. If it relaxes and the diameter of the arteriole is wider, there will be more flow. So the arterioles that perfuse the alveoli, that send blood into the capillaries around each alveoli, those arterioles that perfuse areas where alveolar PO2 is low, they tend to vasoconstrict. They will constrict and be narrower. There will be less blood being delivered to alveolar areas that, remember, you have many alveoli throughout your lungs. If you have an area where PO2 is lower in alveolar space because ventilation into that area is less or inhibited for whatever reason, maybe you have their mucus buildup or some inflammatory process, then the arterioles that feed the blood into that area will constrict and send less blood there. After all, in the end, you want to send the blood to the areas of the lungs, to the alveoli in the lungs that are well ventilated, where there's lots of oxygen. You don't want to really bother as much with areas that aren't. If you look at the arterioles that are serving alveoli where the PO2 is high, they will dilate. They'll widen. You get vasodilation of those alveoli, sending more blood to that area. Again, that makes sense. That's in terms of the arterial vasoconstriction versus vasodilation. That responds to the PO2 level inside the, it's a response to the ventilation of the alveoli. The PCO2, on the other hand, will cause levels of the partial pressure of CO2 inside the alveoli will trigger changes in bronchial diameter. Okay. If you have an area which has a higher PCO2, then bronchioles leading to those areas of the lungs will dilate so that you get good ventilation into those areas, and the CO2 is carried away from those areas, and those serving areas where the ventilation is poor, so that the I'm sorry, wait a minute. I have to think that over again. Hang on. Let me think that over. Yeah, changes in CO2 inside the alveoli will cause changes in the bronchial diameter. If the bronchioles, if you have widening or dilation of the bronchiole, it's because we're talking about an alveolus where there's good perfusion of blood and there's lots of CO2 going into that alveolus. So the bronchiole will have to dilate more to move more air. You need more ventilation in through that area. If the perfusion is poor into that area, then the alveolar PCO2 won't be as high, and you will get constriction of the bronchioles. So PO2 in the alveoli is the key determining factor in arterial diameters, and PCO2 is the key determining factor in bronchial diameters. So you have this coupling of the two, and it's called ventilation perfusion coupling. Blood is sent to those areas that are well ventilated, and air is sent to those areas that are well perfused. And that's the kind of thing that's shown in this diagram. You can go through that. The third key factor determining how efficiently or helping the efficiency of external respiration are the respiratory membranes. Both the surface area and the thickness of the respiratory membrane are key. They need to be thin. They're very thin. That allows for good diffusion of gas. They are only 0.5 to 1 micrometer thick. Just to give you an idea of 1 micron, what 1 micron is, a red blood cell has a diameter of approximately 8 microns. Okay, So if the diameter of the red blood cell is 8 microns, you can see that the respiratory membrane is very thin. They also have a huge surface area, 40 times the surface area of the skin. If you took all the alveoli in the lungs and spread them out flat, you'd get a really large total surface area that allows for exchange of gas. The bigger the surface area, the better the diffusion. The thinner the membrane that the gas has to cross, the better the diffusion. So respiratory membranes are thin and large, both thin and large. Unfortunately, sometimes there are problems. You can get thickening if the lungs become edematous. If you get accumulation of fluid within the lungs and the tissue in there is edematous, and this can occur with inflammatory disease accompanying, for example, infection. In which case, in those areas that have more fluid, you will have inadequate gas exchange, and you could get a deprivation in terms of how much oxygen is getting into the blood. So a person could become oxygen deprived because their lungs are edematous, leading to thickening of the respiratory membrane and lack of exchange, right? If, however, another major problem in terms of respiratory membranes is a decrease in the total surface area of the alveolar surface, and that can occur in a disease called emphysema, when you actually get destruction of large areas, different areas of the alveoli get destroyed, and so you get a reduction in surface area. You get large open areas in the tissue and less diffusion, less possibility for diffusion of gas, oxygen, and CO2 the other way across the membrane. Now let's move from external respiration to internal respiration. Same factors promoting gas exchange that were acting for external respiration act for internal respiration. In the tissues, the PO2 is always less than the PO2 in the arterial blood. Remember that the PO2 in the arterial blood is, of course, a 104, right? And it's always millimeters of mercury. That's left unsaid, but it's 104. And in the tissues, it's going to be 40 or less, depending on how active the tissue is. In the venous blood, 40, right? Because the tissue is 40. So what happens is that the arterial blood brings in the oxygen at a PO2 of 104, and the tissues are at 40, and so you get dumping of oxygen into the tissue, and now the venous blood has the oxygen reduced down to that same level as the tissues. Oxygen was dumped from the arterial blood into the venous, into the tissues, and therefore the venous blood now will be at 40. And but there was uptake of CO2 from the tissues into the blood. The PCO2 of the blood coming in the arteriolar blood was 45, and now has gone to 40. I'm sorry, was 40 and now goes up to 45. So arteriolar blood, I'll write it up here. PCO2 is 40, and the tissues are at 45, so the venous blood becomes 45. All right. PCO2. PO2 goes from 104 to 40. It's not the best. I shouldn't have to write so much on the slide. Anyhow, it's on the previous slides, probably on the next one. Let's see. No, but it was on the previous, on the diagram you have that, and we already talked about it. Okay, in terms of oxygen transport around the body, molecular oxygen is carried by the blood. It's bound to hemoglobin in the blood within the red blood cells. And as you know, each hemoglobin, or should know, each hemoglobin molecule is made up of 4 polypeptide subunits. These are polypeptide strands held together in the hemoglobin molecule by non-covalent bonds, and therefore it's called subunits. Each subunit has within it a heme group, and each heme has an iron at its center. And each iron can take up a molecule of oxygen. The oxygen binds to the heme. And therefore each hemoglobin molecule with 4 hemes can bind, when fully saturated with oxygen, binds a total of 4 molecules of oxygen. A small amount of oxygen also dissolves in the blood, can go just in the plasma itself and dissolve directly in the plasma and be in solution in a dissolved state in the plasma. But vast majority of oxygen is bound to the hemoglobin. Now, the unusual thing about hemoglobin is it has 2 jobs. It needs to be able to take up oxygen, but it also needs to be able to release oxygen. So it has to do binding of oxygen. It has to be reversible, very rapid reversion of the process. This is a term that you won't hear too often, but when hemoglobin is combined with oxygen, technically the term for it is oxyhemoglobin. And if it's has released its hemoglobin, then it's called reduced hemoglobin. Anyhow, here is a diagram. They're both showing hemoglobin. This is a more traditional type of sphere model of the hemoglobin molecule. This is some artist's interpretation of the same molecule. But the idea is that you have 4 polypeptide strands, 2 identical alpha subunits and 2 identical beta subunits, each having a heme at the center of the 4 polypeptides. Hemoglobin, when fully saturated, all 4 heme molecules have oxygen bound. If partially saturated, then anywhere from 1 to 3 hemes are bound up with oxygen. When fully depleted of oxygen, it would be 0 oxygen molecules. The rate, that's speed, in other words, the rate, how fast per time that hemoglobin will bind or release oxygen, is determined, it's regulated by the level of partial pressure of oxygen, the temperature, the pH of the blood. In other words, the concentration. pH is determined by the concentration of hydrogen ions. And in the next few slides, I'll talk about concentration of hydrogen ions as opposed to pH for reasons to simplify things. But you should always realize that as the concentration of hydrogen ions goes up, the square brackets around this H plus indicate concentration. You should realize that as the hydrogen ion concentration rises, blood pH, of course, goes down. And similarly, as blood hydrogen ion concentration drops, pH rises. I'm not going to talk about it in terms of pH. I'm going to talk about it in terms of hydrogen ion concentration. I think it simplifies things. The other thing besides partial pressure of oxygen, temperature, and hydrogen ion concentration is PCO2. And the other thing is, well, we can get rid of this, first of all. That's an old way of saying the molecule's name, di instead of bi. The other key molecule involved in this, or factor involved, is a molecule called 2,3-biphosphoglycerate, or, to make life easier, 2,3-BPG. And we're going to go through each of these in the next few slides. All right. Okay, so this next graph we're going to spend a little time going over. This is the what is called the hemoglobin saturation curve, sometimes called the hemoglobin dissociation curve. What we have here is along this horizontal axis is PO2. And I want to emphasize to you that this is, remember, not a percentage. It's PO2, and that the PO2 in the lungs, in the space of the alveoli, is not 100, it's 104. So this is really pointing at 104. That could better, much better, be a 4. All right. The other thing is the vertical axis, which is the percentage of O2 saturation in the hemoglobin, O2 saturation of the hemoglobin rather. So what percent of the hemoglobin is saturated with oxygen? Now, as you should know, when the blood carrying the hemoglobin in the red cells is in the lungs, it is fully saturated with, it is 100% saturated with oxygen. It can't carry any more oxygen than that. And then it goes back to the heart and gets pumped out into the systemic arterial circulation and travels to the capillary beds in all the tissues where it unloads the oxygen, some of the oxygen that it's carrying. So, and remember, there is no exchange of gases until it gets to that capillary bed of the tissues. So when it gets to the tissues, the hemoglobin is suddenly exposed to surrounding tissue with a PO2 of 40. The blood is carrying a PO2 of 104, and it gets exposed to that tissue PO2 of 40 in the tissues here. So what happens is it will drop or release oxygen because it can't hold on to that fully saturated amount. It can't stay fully saturated, so it releases oxygen. So what you have is a very sudden release of oxygen, and it goes to this level here. Okay, it is not a spectrum like this moving along these different values. It is a sudden exposure to that PO2, surrounding PO2 of 40. Remember, the surrounding PO2 is a key controlling factor in how much oxygen can be carried by the hemoglobin. So then we drop, in terms of percent saturation, what happens is it's dropping from 100% to about 70-75%. Okay. So what you have is now what you have is that it is at this percentage here in terms of saturation. This 100% releases oxygen down to about 70-75%. And you'll see in different books, different figures. It may vary somewhat, and certainly it will change if the tissue is metabolically really active, like muscle tissue that's really contracting a lot, using up a lot of oxygen. But at rest, you're going to get a drop straight down to there. So on this side, and usually this axis is not present in these kind of diagrams, but this shows actually the actual amount of oxygen that gets released. So it's releasing approximately a quarter, right, that's the volume of oxygen that's getting released. I wouldn't worry too much about this on the right-hand side of this axis here. It's usually not presented in that way. It's a question of surrounding PO2, determining how much oxygen the blood can be saturated with. If it's at a PO2 of, if the hemoglobin is in an environment, a PO2 of 104 as it is in the lungs, then the hemoglobin will be 100% saturated. Okay, and if it's in the blood gets delivered to the tissue capillaries, then surrounding PO2 of 40, then you'll get, you know, about roughly 75% saturation. Okay, so that's the hemoglobin dissociation curve, or hemoglobin saturation curve if you want. The influence of PO2 obviously is extremely important. The lower the surrounding PO2, the less the less oxygen hemoglobin can carry, and therefore it'll be less saturated. So we have this oxygen hemoglobin dissociation curve. I left out the oxygen. It's the oxygen hemoglobin dissociation curve. When the blood is in a high PO2 environment, it will be 98 to 100% saturated. It'll be able to carry approximately 20 mils of oxygen for every 100 mils of blood. It's flowing through the capillaries in the lungs where the PO2 is high. It'll pick up oxygen, become highly saturated, anywhere from 98 to 100. As it goes into the systemic circulation and flows into the capillaries in the tissues, then about a quarter of that, remember it drops from 100 to approximately 75, but a quarter of that oxygen is released. So normally in arterial blood, in the systemic circulation, it's completely saturated, and it's not until it gets down to the level of the tissues where it gets exposed to tissue PO2 of 40 will it release the oxygen. The fact that it's completely saturated means that normally when you breathe deeper, you're not going to change the oxygen saturation. It's only when oxygen saturation is lower than fully saturated could you possibly make it more saturated by breathing deeper. If you breathe deeply and ventilate the lungs better, and alveolar PO2 therefore rises, but the hemoglobin is already fully saturated. So yes, in certain situations you could have an effect. Let's say there's a pathological disease process where the person's lungs are not operating, only a small portion, then, or maybe the respiratory membrane is thicker for some reason, then it could have an effect. But otherwise, a normal person when they're breathing, their oxygen will be fully saturated. Even if they breathe deeper, it's not going to increase more. Again, the oxygen hemoglobin dissociation curve showing how much oxygen gets dropped in different conditions. If the tissue that the blood is entering into is highly active metabolically, then it's going to be picking up more oxygen from the blood, because the PO2 of the tissue will be lower. The tissues have used up more of the oxygen to generate energy, and therefore the pressure gradient, partial pressure gradient, is higher, and so more oxygen will be released and go into the tissues. So if you're here, for example, at this value, you know, the curve is quite steep in this area. So if the tissues have a PO2, say a lower PO2 here, then what you see is that as you go, let's change the color, and as you go up and look, you find that in fact because the curve is steep, the curve dives down, then you find, there's quite a bit of change, and it's right here. So you get a lot more oxygen being dumped, even with if you're on the steep part of the curve, then you get a lot more oxygen being dumped, and that's good. So if tissue has lower PO2, then more of the oxygen in the hemoglobin will get released. Exactly what you want. You want to deliver more oxygen to the tissues. Hemoglobin is normally almost completely saturated, in fact, at a PO2 of 70. Now that might not have been too clear on that previous slide, but you'll see in later slides. I mean, it gets up to 95 to 100% even at a PO2 of 70. So we can go to higher elevations where the PO2 is lower than 104 and still get our blood fully saturated with oxygen. The hemoglobin will be able to pick up enough oxygen to be fully saturated. Anything beyond 70, between 70 and 104, you're not going to get that much more increased oxygen binding to the hemoglobin in the lungs. So you get adequate uptake and release to tissues of oxygen even when the atmospheric or alveolar PO2 is lower than 104. Significantly actually lower than 104, and this is a kind of reserve, again, a reserve mechanism that helps. So there's 70, and you can see we're already at 95. There's 70 right there. So when the PO2 is 70, you get, and that's because this oxygen hemoglobin dissociation curve flattens out. It's reaching what's called an asymptote, a leveling off, almost becoming fully saturated even at 70. That's 95% there. We're very close to 95, see? It doesn't make a huge, big difference if you go up in PO2. The gain is relatively small. Very good, which is very good. It means we have a reserve. We can move into an atmosphere which is lower than 104. Humans can function reasonably well at higher elevations. You can't go too high. People have, but they're exceptional. Okay. So normally, in any one single circuit moving around the circulation, the oxygen gets picked up by the hemoglobin in the lungs, goes back to the heart, and you go through one single turn through the systemic circulation. If the person is at rest, not exercising, not doing anything vigorous, then it moves around, and about 20, maybe 25% of the oxygen that's bound to the hemoglobin gets released in the tissues. If, however, the PO2 level in the tissue drops, because, for example, because of tissue's active, metabolically active, and very actively generating energy and needs more oxygen. Remember, oxygen is the final electron acceptor for energy production by aerobic respiration. Then the surrounding PO2 drops more and in the tissues, and therefore the blood coming in will release more of its oxygen. So more O2 dissociates from the hemoglobin and goes into the tissue, the cells in the tissues. In fact, this will happen and deliver quite a bit more oxygen, even if the respiration rate doesn't change and cardiac output doesn't. They don't have to change. There's plenty of oxygen still bound onto the hemoglobin. Remember, normally you're only dropping 20-25% when you're not, at rest. When you're sitting at rest, sleeping at the very low end of usage of oxygen. You only need 20-25%. The blood is returning to the heart through the venous side of the circulation, still carrying about at least 75% of its oxygen that it had. So there's no need actually for respiration rate or cardiac output to change to meet any increased oxygen requirement, because you still have lots more oxygen. At some point, however, depending on how active you are, you do increase the respiration rate, and your cardiac output will change and increase in order to get more blood moving around and going up the lungs and getting picking up more oxygen. But initially, it's it's no big deal. Only a quarter of it. There's still, you have quite a bit of reserve. Okay. There are other factors influencing what the saturation level of the hemoglobin is besides the surrounding PO2. Remember, in the pulmonary capillaries, the goal is to load up with oxygen, pick up oxygen in the lungs, and in the tissues, it's to release oxygen, unload. So this is sort of a contradictory thing, that this one molecule, hemoglobin, can do both. It can pick up with a high affinity, tightly pick up oxygen, and then release oxygen. And it does that because one of the factors is the surrounding PO2. If it's a lower surrounding PO2 in the tissues, it'll release oxygen. But then there are also other important factors involved. For example, all of these. Any one of these can change the affinity, that means how tightly oxygen is bound to hemoglobin. These will all rise in the tissue and body tissues, particularly if it's metabolically active. So all of these tend to rise up in the tissues. It's higher in the tissues, the temperature, the hydrogen ion concentration, the PCO2, the amount of 2,3-BPG in the cells. So any increase actually in these factors will decrease hemoglobin's ability to hold oxygen. It'll decrease the hemoglobin affinity for oxygen, resulting in enhanced or increased oxygen release, unloading from the blood to the tissues. And that's quite good, quite appropriate. After all, when you are exercising, for example, that's the clearest, it's one of the sort of obvious things about metabolically active tissue. When your skeletal muscle is metabolically active, well, you're exercising. It is active because you're exercising, needs a lot of energy. The temperature rises. And with a higher temperature, hemoglobin releases more oxygen. That's good. Your PCO2 is rising because it's more metabolically active. So because of the higher PCO2, more oxygen is released. That's also good. Hydrogen ion concentration is rising. The reason being that with higher CO2 levels, you will get higher hydrogen ion concentration, and that also helps with oxygen release. And this molecule 2,3-BPG in the red cells will rise, and again that helps with unloading. Any decrease in any of these factors, any decreases in these factors will have an opposite effect. Decreases in temperature or hydrogen ion concentration, PCO2 or 2,3-BPG levels will raise the affinity of hemoglobin for oxygen, and so less oxygen will get released from the hemoglobin when it passes through the capillaries and the tissues. So these are all key things. The major one that we discussed and showed earlier was the oxygen hemoglobin dissociation curve, but illustrating the effect of PO2 on hemoglobin saturation with oxygen. Surrounding PO2, if it drops, will cause more oxygen to release from the hemoglobin, but all these other factors are also extremely important. Okay. And just to, talk about it a little bit more, these factors, temperature, PCO2, hydrogen ion concentration to the BPG, your cells that are metabolically more active will be metabolizing more glucose in order to generate energy. And as they metabolize glucose, and it's a process called oxidation of glucose, more CO2 gets released. CO2 is being released in the cells and diffuses into the interstitial fluid and then into the capillaries and into the blood. So CO2 gets released, and that causes an increase in the PCO2 in the blood and also an increased concentration of hydrogen ions. All right. This increasing hydrogen ion concentration, of course, is the same thing and is really the underlying reason why the pH is dropping. Higher hydrogen ion concentration and lower pH. That is acidosis, a lower pH, more hydrogen ion concentration. And that weakens the hemoglobin oxygen bond, the association between hemoglobin and oxygen. So the hemoglobin can release more oxygen, which is good. That's when you do want to have more oxygen delivered to the cells when they are metabolically more active. So this effect of pH, in other words, hydrogen ion concentration, is called the Bohr effect, B-O-H-R. And I'll show you on a graph the Bohr effect, what the Bohr effect, the effect of I'll show you the result of changes in pH on hemoglobin, on the oxygen hemoglobin dissociation curve. The other thing is that metabolically active cells produce heat. The more metabolically active they are, the more heat they produce. When we form energy by the oxidation of glucose, the breakdown of glucose, it's not 100% efficient. All the energy does not, from the carbohydrate glucose, does not go to ATP production. Some of it results in, of that energy is generated as heat. And so you get a rise in temperature. You know that when you're physically active, you have a rise in temperature. And a rise in temperature will trigger a very rapid increase in 2,3-BPG synthesis inside the red cells. So all of these factors, temperature, hydrogen ion concentration, and others, pH, PCO2, and 2,3-BPG, in fact, are higher in the systemic capillaries, where O2 unloading is the goal after all. And so O2 is ensured, O2 unloading is ensured in the vicinity of tissue cells that are metabolically active and working. Here you see the normal body temperature, 37, 38 degrees. That's normal body temperature of blood. Unfortunately, in this graph, they've drawn it and they didn't show full saturation up to close to 100%. 40, 50, 60, 70, 80, 90, 100, 104 would be here. No, I'm sorry. 104 is here. They're not showing 100%. Some of these graphs are, on this side, they are showing it coming up to 100%. I'm not sure why, but that's not the main point of the graph. The main point of the graph is to illustrate the effect of body temperature on saturation in this oxygen hemoglobin saturation curve. What you have is it clearly shows the shape of the curve changes with increasing body temperature. So you get a steeper curve coming down at a higher temperature, and therefore less oxygen. In fact, at any, at the PO2 in the body, you see that this much less oxygen is held, so that more oxygen is getting released. The opposite is occurring when, as the temperature drops. As the temperature drops, hemoglobin has a higher affinity for oxygen, and that's shown up here at 20 degrees. Even if the PO2, surrounding PO2 is lower, it's at the tissue level, it's holding on quite a bit to oxygen. It started here and releasing here, but body temperature of 20 is extremely, core body temperature of 20 degrees is extremely low. The graph on the right side is showing the effect of PCO2 on the shape of the curve. Normal PCO2, of course, is sorry, just a second. Normal PCO2, normal arterial PCO2 is 40, normal hydrogen ion concentration, and therefore normal pH is 7.4 in normal arterial blood. And when it goes into the tissues, it goes up. If the tissue is particularly active, you could have remember, tissue PCO2 is normally at rest, say 45, but it could go up quite a bit, and causing also the pH to drop. And that causes this kind of shift again to the lower right, a shift over in the curve towards the lower right in this direction. And so more oxygen is being released.
And we're going to stop here. This is the end of Unit 5, Part 3.
The characteristics of how tightly oxygen can be held onto oxygen is held onto by hemoglobin.